Hello, my name is Min,
I'm Alex.
I'm Sophia.
And I'm Jacqueline.
and we're from the Department of Microbiology, Immunology, and Molecular Genetics at UCLA. And our project is titled A Relational Analysis of Rhodococcus phage clusters and singletons. Bacteriophages can be characterized by their isolation host, and some examples of this include Mycobacteria, Rhodococci, and Gordonia. Our group decided to look at bacteriophages isolated from Rhodococcus bacteria. As a part of the Actinobacteria phylum, it is highly similar to Gordonia. Interestingly, a large percentage of sequenced and fully annotated Rhodococcus phage genomes are singletons. Also, these singletons make up 19% of all known Actinobacteriophage singletons, which is surprising because Rhodococcus phages make up a small percentage of all known Actinobacteriophages. Therefore, our goal was to explore the relationship between these Rhodococcus singletons and other phages. We took phage genomes that were sequenced, assembled, and annotated by various other institutions and performed bioinformatic comparison analyses on 3 different levels: whole genome, single gene, and phage to phage. Some tools we used were nucleotide and amino acid sequence comparisons, genome content similarity analysis, nucleotide and gene content similarity visualization, and finally split strain networks based on shared genes.
First, we analyzed the genome metrics of all Syphoviridae morphotype Rhodococcus phages available on PhagesDB. We looked at genome length and nucleotide composition, which is measured here in GC%. We found a significant downward trend here, suggesting that there may be some pressure for longer phages to use less GC base pairs, possibly because this is more energetically costly. This may also suggest that CB and CE are more distantly related to the other phages. Next, we looked at GCS. Singletons have no known close relatives, so their GCS are expected to be low. However, we find that some singletons share some similarity. REC2 and WAC, for example, share 31% of FAMs, which are high for singletons, and which we will explore later. Most Rhodococcus singletons share little similarity with each other, but their whole genome trends are reflected in their single genes.
We then looked at conserved single genes that are relevant for replication and assembly. Again, we see the inverse relationship between the clusters and similarity between the singletons REC2 and WAC. The terminase and portal subunits form their own individual complexes that work together in capsid assembly and share similar patterns of relatedness in their dot plots, with notably high intercluster similarity between clusters CB and CE. The tape measure protein is a scaffold for tail assembly and consists of highly conserved tandem repeats, which makes it a candidate for determining cluster and other actinobacteriophages. This is not the case in Rhodococcus phages, as there is weak relationship within cluster CE, despite the high intercluster similarity throughout. Lastly, we looked at lysine proteins that break down the host cell wall and observed wide intercluster similarity across clustered phages and singletons. This is believed to be due to the phage's preferred host, which can differ from its isolation host.
After whole genome and single gene analysis of Rhodococcus clusters and singletons, we still see that there are not a lot of similarities between the singletons and clusters. From there, we wanted to expand our analysis to include Gordonia phages because Gordonia and Rhodococcus hosts are similar. In panel A, the distance on the split tree indicates gene content similarity, and we can see that Rhodococcus singletons and Gordonia phages are grouped closely together. We have DocB7 with beaver, EpicDab and Coer with RRH1, and we also have 2 Rhodococcus singletons, REC2 and WAC. The GCS percentages for these pairings range from 29 to over 50%. Panels on the right show the FAM maps correlating with the groups circled in the splits tree. We can observe alignment of FAMs between the phages as shown by these boxes for structural and other genes, and this is what is called synteny. We also see nucleotide sequence similarity between REC2 and WAC, and some similarity between Coer and RRH1. This information could be used to help us make inferences of evolutionary relationships between these phages, whether it is convergent evolution, where phages are evolving similar functions to infect similar hosts, or other evolutionary mechanisms.
In summary, after we verified previous findings of inverse intercluster and intracluster relationships between whole genomes of Rhodococcus phages, We found that generally these same patterns were in single gene comparisons with some notable differences. Also, we found that there were considerable similarities between Rhodococcus singletons and Gordonia clustered phages, suggesting the need to study phages outside of their assigned clusters or isolation hosts. Thank you.